Okay, we're back for lecture 2. Hopefully this isn't going to take, I have 12 more slides to get through. Anyway, so what we're talking about here is a sire that's heterozygous, meaning his genes carry DNA for the same trait, but different qualities of the same trait. So this would be coat color. And so we ended up saying the phenotype is what the animal looks like, which is three quarters, 75% of the time the calf would be born with black, being black hided, and then 25% of the time he would be born having a red hide. So this sometimes is like, considered, oh, well, he must not have got bred to the correct bull. Well, that's because both the sire and the dam have to be carriers in order for that recessive trait to show up. So if we look at the genotype, which is what the genes say 25% homozygous dominant, 50% heterozygous, and 25% homozygous recessive. Okay, so let's talk about quantitative traits. So quantitative traits are traits that can be measured objectively. And remember, in a lot of animal genetics, the traits are one gene doesn't control a complicated trait. So if you're looking at feed conversion or behavior, in horses you have weight, height, speed, these traits are controlled by many genes, not just one single gene like coat color in cattle. So qualitative traits are ones that are controlled by a little bit, a smaller number of genes. So we have coat color in cattle, the trait of having horns versus being born without having horns, that's called polled. So that's being controlled by a single gene pair. So sometimes you don't have the trait, so genes can interact. You don't always have the trait where you have the black coat color covers up the red coat color. So if you have a dominant bull with a black hide, a homozygous bull, all his offspring, 100% of them, are going to be black, have a black coat. Sometimes you have, that's called complete dominance. And so the homozygote and the heterozygote would both have a black hide. They would both have the same phenotype. And the recessive, which is only 25% chance, depending on how they're bred, has the lowest phenotypic value. So those are the ones you don't want. And it also shows that both parents have to be a carrier. And then sometimes you have what's called incomplete dominance. And incomplete dominance or lack of dominance is when you get a blending of the trait. So like I told you, when we breed some kinds of cattle, like we breed a black Angus to a Charolais, which has a white coat color, you would think that black would overpower it. It doesn't. Only works in the English breeds. In the continental breeds, we breed a black Angus to a Charolais, you get a brown calf. So you get blending. And then over dominance is like hybrid vigor, where you have the heterozygote, this one performs better than this one or this one, the homozygote recessive or the homozygous dominant. That is called hybrid vigor. And that is the reason why we crossbreed a lot of livestock, because we want this overdominance trait, because it works in a feedlot. Crossbred cattle have better feed conversion than straight bred cattle. Crossbred cattle are more resistant to disease, and it's true for crossbred pigs and all livestock species. So that combination of two breeds creates that heterozygote. Okay, so here's just a picture we were talking about cows with red coats and black coats. And these could be Angus cattle. So Angus cattle, you have red Angus and black Angus. So the black Angus has to carry the black gene. These little guys could be the result of from a red mom bred to a black bull. You could get a black colored calf. Or it could be from a mom that carries the homozygote black gene. Another trait that we see a lot of times in cattle is a trait of horns or being born polled. So it's not called hornless, it's called polled. So it just so happens in cattle, this is kind of weird, but in cattle, the horned trait is the recessive trait and the polled gene is dominant. So polled cattle are either homozygous for polled, those would be purebred cattle, or heterozygous, meaning they have horns. So for the most part in cattle, we don't want them to have horns. If they're out on the range like this guy looks like he is, out in Wyoming or Colorado or something, then having horns might save you from getting eaten by a bear. The problem with horns is in a feedlot, you just gore, your other friends in the feedlot with you. So a lot of times cattle have to be dehorned. And that's a painful process and it sets the cattle back. And so rather than dehorning them, sometimes we breed to animals that are polled. So polled is dominant. So this is the same breed. These are both Herefords. And one has horns and one doesn't. This is a polled Hereford and the other one is a horned Hereford. So the polled cattle pass that trait on. A lot of the continental breeds, like Maine Anjou and Simmental, all of those breeds are horned. Angus cattle are naturally polled. So when you bring in the Angus bull, you are naturally dehorning your cattle if he's homozygous dominant for polled. And it saves time, it saves money, and it saves, that growing of the cattle because they don't have to be dehorned. Okay, so this is just a graph showing I'm not really sure what it's showing, the value of different phenotypes. So the value in complete dominance, the value we are breeding for, we want that dominant trait. So a lot of times recessive traits are code for things that aren't necessarily good. Really, coat color is not a big deal. It doesn't really matter. But it's easy to study in genetics. So we want this dominant trait. When you have incomplete dominance or lack of dominance, this AA is a blending of this one and this one, so it has less value. But a lot of times recessive genes can be genes that aren't good. They can code for, negative traits that might lower production. And then this one, I know you can't see because I'm in the way here. This one is when we have a hybrid vigor, which you have the crossbreed having the greatest genetic value. Okay, and then this is just a picture of a swine egg that is being injected in the nucleus, micro-injected with somatic cells or new genetic material. So that is the process for cloning. So some information on biotechnology. This is kind of interesting that they even talk about it, but I just think they picked kind of a really weird example. But biotechnology is the use of living organisms to improve, modify, or produce industrial important products. So a lot of times we use bacteria, and we use bacteria to create lots of kinds of foods that we eat. So yogurt, we have the acidophilus bacteria. In cheese, we have cheese cultures that, the cheese manufacturers are really proud of for their fancy French cheeses and things like that, or Dutch cheeses. But those microorganisms are introduced to cause fermentation or some other process. So it's bacteria. So we can use these same bacteria to help us create animal products that we consider to be important. One of the things that we have available to dairy farmers is called BST. BST is bovine somatotropin. That's what BST stands for. Bovine somatotropin is a hormone that it can be given to dairy cows, and it increases their milk production by about 10%. The hormone is naturally occurring in beef cattle. So we can boost that naturally occurring hormone by giving them artificially manufactured BST. This is very controversial. The little r here stands for recombinant. When it's recombinant, we're using microorganisms to produce the BST. So this is a bovine hormone, but we're using recombinant DNA techniques, using bacteria to help produce this hormone, which can then be injected into dairy cows, and they can increase their milk production 10%. So that is a controversial issue. And if you look on a carton of milk, especially in California, or a carton of cottage cheese or yogurt or whatever, a lot of times it'll say milk from cows not treated with RBST. So it's very controversial because people felt like they were giving artificial hormones to beef and dairy cattle, in particular dairy cattle. So this was huge, and it was very bad PR for the dairy industry for a while there. So that's why that RBST is not a real great example for the livestock industry. And this just shows you how RBST is created, and it can be given to an animal. And it's, like I said, it's a naturally occurring bovine growth hormone. It's injected into lactating cows. It helps direct the energy to feed to meet the animal's need for growth and milk production. And it increases milk consumption and feed consumption and milk production by about 10 to 20%. So, this book is a little bit old, but mapping the animal genome, it has been done for many animal species. I know it's completely done for poultry, so we know what each gene codes for. And that helps us to identify traits that we want and traits that are desirable and traits that are undesirable. And in the big picture, that's where genetics becomes controversial, but at the same time, very important, because it allows us to select for disease resistance, improve fertility, all the things that help us make animals more efficient in their productivity. It also can allow us to produce animal drugs that can target specific genes to turn on or off. And then we already talked a little bit about cloning in animal genetics in the last chapter. Okay, well, that is chapter 12, a little complicated. I hope you had a chance to read it over in your book. All right, thanks.